So my presentation was about the relationship between obesity, inflammation and diabetes. We know that there's a large increase in the number of obese individuals worldwide, and this is associated with what we call low-grade inflammation, the expression of cytokines in various metabolically active tissues, and that these cytokines are responsible in many ways for the development of insulin resistance, which is crucial for development of type 2 diabetes. However, we should also remember that in many individuals that do become obese and insulin resistant do never develop type 2 diabetes. And that is because their pancreatic beta cells, the insulin producing cells in the pancreas, are able to compensate for the insulin resistance, increasing in number and increasing in activity. So we are trying to understand what are the mechanisms at the cellular and molecular level between the individuals that do not become diabetes but are obese and the individuals that do develop diabetes in response to obesity. So we have looked at a number of factors and pathways that are responsible for upregulating this beta cell activity in normal obesity and have focused on a class of molecules known as the BMPs, the bone morphogenetic proteins, and how they might affect the beta cell function. So we found that they are actually upregulated in the pancreas in response to inflammation, in response to cytokines, and thereby act as inhibitors of the compensation, inhibitors of proliferation and inhibitors of beta cell function. If we apply now compounds that can actually inhibit the effect of the BMPs, then we regain the ability of the beta cell to compensate, both in terms of upregulating the number by proliferation of beta cells, but also by upregulating the activity, the insulin secretion in response to glucose. So while all these studies have been done in vitro, we are now moving towards the in vivo situation where we in animal models would like to see if similar principles apply there in different diabetes models to see if we can prevent or maybe even treat type 2 diabetes by inhibiting the effects of these BMPs.